Good evening, everybody. Welcome to the 11th session of the weekly interactive course on Genome Editing and Engineering, taken by Professor Utpal Bora of IIT Guwahati. I'm Sanket Mukherjee, Prime Minister's Research Fellow from the School of Biological Sciences, IIT Delhi. Let's look at today's slides. So, Question 1. Ayurveda classifies all individuals into different prakriti based on the theory of A. Vata, B. Pitta, C. Kapha, or D. Tridosha. Is to talk about the history of personalized medicine in India. Ayurveda is a national healthcare system that dates back more than 5,000 years in India. It thinks that each individual is unique and has particular constitution and hence emphasizes on personalized treatment. Ayurveda classifies all individuals into different prakriti based on the theory of three dosha. This is independent of racial, ethnic, or geographical considerations and may provide appropriate means of classifying phenotypes to be considered collectively for genotyping. If we talk about the history, the three doshas are Vata, Pitta, and Kapha, which is space, air, fire, water, and earth, are the five essential components of life that exist in human body as three primary humors known as the three doshas. The component of space and air combine to form the Vata dosha, and Pitta dosha is made up of the elements fire and water. The Kapha dosha is made derived from earth and water components. Ayurveda categorizes drugs according to Ras Panchaka, Ayurvedic pharmacology, which states that the drug's action is attributed to certain factors present in the drug as follows. Find the wrong combination. Is it rasa, state, taste, guna, effect, virya, potency, or vipaka, post-digestive taste? So if we talk about the history, it categorizes drugs, Ayurveda categorizes drugs according to Ras Panchaka, Ayurvedic pharmacology, which states that the drug's action is attributed to certain factors present in the drug, namely the ras or the taste, gun or property, virya or potency, vipaka or post-digestive taste, and prabhav, that is effect. Whereas modern pharmacology attributes the drug action to chemical structure of a molecule. The Ras Panchaka modality can deliver treatment because it considers the person's prakriti as well as the pharmacodynamics and pharmacokinetic properties of a drug, as opposed to modern treatment, which elicits different response from person to person, even when using the same drug for the same disease. Garrett is best known for his book Inborn Errors of Metabolism, where he claimed that four diseases, alkaptonuria, albinism, cystinuria, and pentosuria, were inherited as Mendelian autosomal recessive trait and was published in the year 1909, 1902, 1809, or 1990. So Archibald D. Garrett and Personalized Medicine, if we talk in 1902, he published the paper, The Incidence of Alkaptonuria, a study in chemical individuality, where he suggested that alkaptonuria is not the manifestation of a disease, but is rather of the nature of an alternative course of metabolism. He is best known for his book, Inborn Errors of Metabolism, published in 1909, where he claimed that four diseases, alkaptonuria, albinism, cystinuria, and pentosuria, were inherited as Mendelian autosomal recessive trait. The foresightful effort paved the way for the study of hereditary illness and established Garrett's status as the father of medical genetics. We talk about the timeline of modern medicine. So the prehistoric medicine, it dates back to 8000 BC. Ancient Egyptian medicine, it dates back to 2900 BC. Ancient Chinese medicine dates back to 2700 BC. Babylonian medicine at 2300 BC. Ancient Indian medicine at around 1500 BC. Greek and Roman medicines from 1200 BC to 300 AD. Arabic medicines in 16th and 17th century, and the modern medicine in the 20th century. In 1926, pamaquine and 8-aminoquinoline was one of the most effective medications used to treat A, AIDS, B, cancer, C, dysentery, or D, acute malaria. A quinoline group of compounds are usually used to treat malaria. Malaria was first recorded in ancient Chinese medical archives in 2700 BC. Even today, malaria is considered as one of the most serious and lethal disease. Several plants, including quinjai and in the 22nd century BC in China and the cinchona tree in the 16th century in Peru, have been used to cure malaria. Pamaquine and 8-aminoquinoline was one of the most effective medications used to treat malaria in 1926. However, administration of pamaquine showed the adverse effect of hemolytic anemia in many patients. As a result, scientists began researching alternate therapy to combat the side effects of this drug. In the Korean War, primaquine and 8,4-amino 1-methyl-butyl amino 6-methylquinoline was used as an anti-malarial drug to eliminate the long latency of Plasmodium vivax infections of the soldiers. In 1956, Carson et al. Discovered that the side effect of hemolytic anemia were caused by a deficiency in glucose 6-phosphate dehydrogenase enzyme. The discovery of the connection between anti-malarial drugs and the glucose 6-phosphate dehydrogenase enzyme led to a new perspective on the adverse effects of these drugs, as well as a more personalized approach towards the disease, which was one of the first examples that led to a big step towards the application of a more personalized therapy. So this is the timeline of personalized medicine. As you can see, Shendia's group published the first proof of concept that NGS technology may be used to diagnose genetic diseases in September 2009. After a few months, they reported Miller syndrome, the first recessive disorder through whole exome sequencing, whole genome sequencing, whole proteome sequencing, or whole plasmid sequencing. So next generation sequencing and personalized medicine. So next generation sequencing technologies have helped in the development of personalized medicine by providing scientists with a strong tool to comprehend each patient's condition and its unique genetic features and whole genome mutational status with single nucleotide resolution. The Human Genome Project, it completed in 2001, opened the door to a better knowledge of precision medicine. So this is how it came off, the history of sequencing technology. And these are the steps which are used for sequencing, which you can read about, right? So Shendia's group, they published the first proof of concept that NGS technology may be used to diagnose genetic diseases in September 2009. After a few months, they reported Miller syndrome, the first recessive disorder through whole exome sequencing. Well, engineering smart biomaterials for drug delivery systems that can detect the demands of a person's physiological condition and change their drug release profile accordingly is a clean method for creating a precision immunization, precision pharmacokinetics, pluripotent stem cells, or precision biomaterials. So when we're talking about biomaterials for precision medicine, the advances in this field have enabled creative product designs to target and cure diverse ailments that may be required to meet the rising need for precision medicine. Precision biomaterials are engineered with specific mechanical and biochemical features to enable the design of personalized implants or organ replacements. Precision biomaterials must be designed to perform a variety of unit operations, such as separators, which can specifically attach to molecules, or cells for separation, sensors, which can detect particular analytes, or electrochemical signals or responders, which can change their morphological features, or degrade, and controllers, which can change the material properties to release the payload. Engineering biomaterials. So in addition to supporting the growth of artificial tissues, biomaterials can be specifically designed for direct in vivo tissue manipulation. Biomaterials could also help in enhancing the efficacy of precision medicine through the engineering of smart drug delivery systems, through pharmacokinetics, drug delivery system to foster the accuracy of precision medicine. So engineering smart biomaterials for drug delivery system that can detect the demands of a person's physiological condition and change their drug release profile accordingly is an appealing method for creating precision pharmacokinetics. So similarly, we can engineer biomaterials by having carriers with gene treatments such as plasmid DNA, mRNA, interfering RNA or microRNAs. The formulation of the nanomaterial may be customized to match each patient's unique needs based on their unique genetics. So DASH are one class of smart polymers that can repair their structure after repeated injury and are frequently injectable by needles. Is it polymeric biomaterials, miRNA polymers, precision biomaterials, or self-healing polymers? Obviously, it would be the self-healing polymers. So there are smart polymers that are one type of soft material that respond to environmental changes. The thermally sensitive polymers, which are frequently employed as cell carriers and in 3D printing, are good examples. Self-healing polymers are one class of smart polymers that can repair their structure after repeated injury and are frequently injectable by needles. Another type of material with the capacity to remember its original shape is shape memory polymers. These smart polymers can transport proteins, drugs, and cells. They can be used in precision medicine, minimally invasive surgery, and bioprinting thanks to their injectability and shape memory capabilities. The smart polymers, if you see, a summary of the process is given in the diagram. The term theranostics, which was first used Funkhauser, refers to scientific advancements that aim to create more specialized and personalized treatments for a variety of A, organoid conditions, B, animal model conditions, C, pathological conditions, or D, humanoid conditions. Theranostic is a term used for a combination of therapeutics and diagnostics. The term theranostics, which was first used by John Funkhauser, refers to scientific advancements that aim to create more specialized and personalized treatments for a variety of pathological conditions. These advancements also aim to combine diagnostic and therapeutic applications into a single agent, resulting in a promising therapeutic paradigm that includes diagnosis, drug delivery, and treatment response monitoring. It provides a diagnostic therapy for specific patients, testing them for potential adverse drug reactions, and then customizing a course of action for them depending on the test results. Question 9, which of the following about precision biomaterial and operation is not correctly matched? Is it A, separators, which can specifically attach to molecules or cells for separation, sensors, which can detect particular analytes or electrochemical signals, responders, which can change their properties to release the payload, controllers, which can change the material's property to release the payload? I'll give you a minute to think on this and then answer. C, responders, which can change their properties to release the payload. Which of the following is a lipid-based drug delivery system? Is it liposome, silica nanoparticle, dendrimer, or polymer micelle? The liposome is the answer. What are self-healing polymers? I think we already discussed these. Smart polymers that has the capacity to remember its original shape. Smart polymers that can repair the structures after repeated injury and are frequently injectable by needles. Frequently employed as cell carriers and in 3D bioprinting. And all of these. So the answer would be B. Small polymers that can repair the structure after repeated injury and are frequently injectable by needles. Which technology is used for precision medicine? Is it smart polymers for cell therapy, smart devices, organ on a chip, or all of the above? All of the above is the answer. What is created? The VXDS program, who created it and when? So voluminous exploratory data submissions, a repository for genetic information created by FDA in 2003. Voluntary exploratory data submission, a repository for genetic information created by FDA in 2013. Voluntary exploratory data submissions, a repository for genetic information created by FDA in 2003. Or voluntary experimental data submissions, a repository for genetic information created by FDA in 2003. So it would be voluntary exploratory data submission, a repository for genetic information created by FDA in 2003. Which of the following is a potential issue associated with long-term expression of CRISPR enzyme delivered via the adeno-associated virus in vivo clinical studies? Is it the reduced therapeutic efficacy, limited gene targeting, unintended genetic mutations, or inadequate delivery system? So the answer would be unintended genetic mutations. What is the full form of UMIs? Is it unique molecular identifiers, uniform molecular identifiers, unique modification identifiers, or uniform modification identifiers? The unique molecular identifiers. Why CRISPR unique molecular identifier technique is more suited to explore the underlying heterogeneity of cancer phenotypes? A, because it statistically normalizes the effect of clone dynamics on relative abundance of guide DNAs. B, it statistically normalizes the effect of genomic dynamics on relative abundance of guide RNAs. Or it statistically normalizes the effect of clone dynamics on relative abundance of guide RNAs. Or is it D, none of the above? So the answer would be C, statistically normalize the effect of clone dynamics and relative abundance of guide RNAs. Which of the following statement is true regarding therapeutic decision based on genetics? A, gene variations have no impact on pharmacological reactions in patients. B, therapeutic decision based on genetics are solely determined by multi-gene analysis. C, gene variations impacting drug response can lead to modifying dosage or changing treatment. Or D, single nucleotide polymorphism profiles are not considered in evaluating gene variations. Answer would be C. Gene variations impacting drug response can lead to modifying dosage or changing treatment. Question 18, which is the last question of the day. What is the primary focus of pharmacogenetics? Is it A, researching drug manufacturing processes, B, investigating drug interactions with food, C, studying individuals' reaction to drugs, or D, analyzing drug distribution in the body? It's C, studying individuals' reaction to drugs. So with that, I think we are over with today's session, and we'll be meeting next week, same time, 7 to 9 on a Tuesday for the week 12, the final week of this course. If there are any questions, now is the time to ask. If there are none, then I would call it a day and I'll stop the recording.